All right, so if you're in my psychology of learning class or just want to know more about classical conditioning, we're going to go through some of the theories and research on classical conditioning in this PowerPoint presentation. The main ones that we're going to focus on for this presentation would be the blocking effect, the Rescorla-Wagner model, theories of attention, and then comparator theories of conditioning. When we look at the blocking effect, we look at this as a phenomenon in classical conditioning. This is where a previously learned association between a stimulus, or a conditioned stimulus, and an unconditioned stimulus prevents the formation of a new association with a second stimulus when two stimuli are presented together. Essentially, the organism blocks the new association because it already has a strong predictive relationship with the conditioned stimulus and the unconditioned stimulus. So, for example, imagine a rat is conditioned to associate a light, which be the conditioned stimulus, the first one, with a shock, the unconditioned stimulus. So every time the light comes on, the rat expects a shock. Later, we introduce a second conditioned stimulus. This would be the CS2. This is introduced alongside the light, but the tone doesn't provide any new information about when the shock will occur. The light is still the predictor. So as a result, the rat does not learn to associate the tone with the shock because the light already serves as a reliable signal, and this is the blocking effect. The prior learning of the light-shock association blocks the learning of the tone-shock association. So this model is a mathematical model that's used to explain the process of classical conditioning. It describes how the strength of an association between a conditioned stimulus and an unconditioned stimulus change over time based on the discrepancy between expected and actual outcomes. The model's key idea is that learning occurs when there is a surprising or unexpected outcome. And so this model has 6 fundamental rules. The first one and second that you see here, the first one, learning occurs when the outcome is surprising. The central concept of the Rescorla-Wagner model is that learning happens when the unconditioned stimulus is surprising or unexpected. If the US, the unconditioned stimulus, is already fully predicted by the CS, little to no learning takes place. Learning is driven by the difference between the expected and the actual outcome, known as the prediction error. The second one, strengthening of association, occurs when the actual outcome is greater than expected. I'm going to say that one more time. Strengthening of association occurs when the actual outcome is greater than expected. When a conditioned stimulus, a CS, predicts a US unconditioned stimulus, but the US is stronger or more intense than expected, the association between the CS and the US strengthens. This happens because the organism is surprised by the stronger than expected outcome, leading to increased learning. So 3 and 4 here, 3 is weakening of associations occurs when the actual outcome is less than expected. So what that means is if the expected unconditioned stimulus does not occur, or if it's weaker than expected, the association between the CS and the US weakens, and the organism is surprised by the absence or the lower intensity of the US, and as a result, the strength of the conditioned response will decrease. For number 4, the learning or no learning occurs when the actual outcome matches expectations. And what that means is when the predicted US unconditioned stimulus perfectly matches the actual US, there's no prediction error, no further learning takes place. This is because the CS fully predicts the outcome and the organism is not surprised. So the associative strength remains stable. Number 5 is salience of the stimuli affects the rate of learning. The rate of learning is influenced by the salience or the intensity of the CS and the US. More noticeable or attention grabbing leads to faster learning. For example, a loud sound may result in faster conditioning compared to a soft sound because the organism pays more attention to the louder stimulus. The total associative strength of all stimuli is limited. The model assumes that there is, that's number 6, I'm sorry, I kind of jumped right into that, but so the number 6, the total associative strength of all stimuli is limited. So the model assumes that there is a limit to the total associative strength that can be learned for a given US unconditioned stimulus. Once the organism has learned to fully predict the US from one or more CSs, then no further learning can occur. And the principle explains the phenomenon like blocking, like we discussed a few slides ago, where the presence of an already well-established CS can prevent new learning about additional stimuli because the associative strength limit has been reached. So this is the model's core formula in terms of mathematics. And key concepts of this model include prediction error, blocking, and extinction. So prediction error is learning that is proportional to the size of the prediction error and the difference between the expected unconditioned stimulus and the actual unconditioned stimulus. So we're looking at the difference between those two. Blocking, so the model explains the blocking effect where a conditioned stimulus that already predicts the unconditioned stimulus prevents learning about another conditioned stimulus that's presented at the same time. So like that CS1 and CS2, that's what that's referring to. The CS1 is already there, so it's not going to learn the CS2. And then extinction, the model accounts for extinction which occurs when the CS is presented without the UCS or US unconditioned stimulus, causing the associative strength to gradually decrease. So let's look at this example. Imagine you're conditioning a dog to associate a bell with food, right? Pavlov's experiment. Early on, the dog learns this association quickly because the food is surprising. But over time, as the dog predicts the food perfectly, the learning rate slows down. And then if you introduce a light alongside the bell, the dog won't learn much about the light if the bell already fully predicts the food. This is illustrating blocking. If you stop providing the food after ringing the bell, the dog's response to the bell will gradually diminish, which is extinction. So this model provides a framework for understanding the dynamics of learning, particularly in situations where multiple stimuli are involved or where the intensity of stimuli can vary. The blocking effect can be explained through the model of conditioning, which states that learning occurs when there is a discrepancy between expected and actual outcomes. Since the CS1 already predicts the outcome, which is the shock, the CS2 does not add any new information, so no learning occurs for the CS2. So let's look at an example again. A practical example of the blocking effect can be seen in marketing. If a brand already has a strong association with a certain product, say brand A is known for high quality laptops, introducing a new feature like a touch screen alongside the established reputation of high quality may not form a strong association in the consumer's mind. The pre-existing belief of high quality blocks the new association for a touchscreen feature, making it harder for the feature to stand out. Overall, the blocking effect highlights how prior learning can influence new learning, and particularly when no additional information or surprise is introduced by the new stimulus. Looking at some other theories of associative learning, we have the theories of attention and the comparator theories. In classical conditioning and learning theories, animals and humans are thought to direct their attention to stimuli that are informative and that they predict meaningful outcomes and ignore those that are uninformative or do not predict outcomes. This reflects the idea that animals optimize their behavior by focusing on the cues that help them understand their environment, which enhances survival and efficiency. So there's a lot of other theories, and I'm just going to briefly mention some of these. But McIntosh's theory of attention is one. And the key idea here is the amount of attention an animal gives to a conditioned stimulus depends on how well that stimulus predicts an unconditioned stimulus. Stimulus that reliably predict outcomes receive more attention, while those that fail to do so get ignored. So for example, if a tone consistently predicts food, the animal will focus on the tone. But if a light is presented at random without any consequence, the animal will ignore the light over time. And then we have Peirce-Hall's model from 1980. The McIntosh was from 1975, but Peirce-Hall is from 1980. And this, key idea is that attention is given to stimuli based on how surprising or uncertain the outcome is. Attention decreases when the outcome becomes fully predictable. An example here is early in learning, when the animal is unsure if the tone predicts food, it will pay a lot of attention to the tone. But as the animal becomes confident that the tone always predicts food, then its attention to the tone decreases because the outcome is no longer surprising. And, I've said this in other slides and in classes and stuff, but um, if we look at Skinner and operant conditioning, like one of the things that he says is that the reward itself is not as um, it's not as much of a reinforcer than the hope, right? It's the hope that something's going to happen. So if something becomes too predictable here, then you're not getting the same effect as if, it's more randomized when you present stimuli to it, right? So it's that surprise, that hope that something is going to happen that keeps people or animals more motivated than the actual reward itself, as you can see here in the examples. So aplysia is research that we've done that provides strong evidence for growth of the new synapses. It's a process that's crucial for long-term memory storage. So long-term sensitization in aplysia leads to the formation of new synapses between sensory and motor neurons. The synaptic growth is not just a chemical change, but also a structural one, where new connections are formed, physically formed between neurons, which strengthens the communication between them. Structural changes that we see in addition to the new synapse formation, long-term sensitization in aplysia also results in branching of dendrites and the enlargement of existing synapses. So these physical changes in the neuron structure provide a more robust neural network, which enhances the ability of the animal to remember and respond to stimuli over extended periods of time. The synaptic plasticity is the process of adding new synapses, and then that strengthens the existing ones, which is the ability of synapses to change in strength and structure. And so this plasticity is the foundation that we have for learning and memory. Research here provides clear evidence for both chemical changes and growth of new synapses during learning and memory formation. Chemical changes such as the modification of neurotransmitter release through molecular cascades are crucial for short-term memory. In contrast, the growth of the new synapses regulated by gene expression and protein synthesis underlies long-term memory. And these findings that we see have been foundational for understanding the biological basis of learning and memory in more complex organisms such as us, humans. The contiguity principle, as well as the taste aversion, is what we cover here. And so the contiguity principle suggests that for learning to occur, a stimulus and a response, or two stimuli, must be closely associated in time. So in classical conditioning, it's believed that the closer conditioned stimulus and the unconditioned stimulus are presented together in time, the more likely conditioning will occur, right? So we present them simultaneously. Application in taste aversion learning, this deviates a little bit. So taste aversion learning deviates from the principle. In taste aversion learning, an animal can learn to associate a particular taste with illness, even if the sickness occurs hours after consuming the food. And this goes against the contiguity principle because typically a long delay between the CS and the US should weaken the association, but in taste aversion learning, this association can still be strong. And this is where we get like food poisoning. Like when you eat something, there's no immediate, right? You're not pairing that CS and UCS, or reaction response together right away. There's a delay in sickness, right? So that's your taste aversion learning, which goes against the contiguity principle. And so we see this taste aversion happening in cases of like foodborne illness or food poisoning, something like that. There's a biological preparedness here in taste aversion learning that refers to the idea that organisms are evolutionary predisposed to form certain associations more easily than others due to survival advantages. In the context of taste aversion learning, animals, including humans, are biologically prepared to associate certain stimuli like taste with negative experiences like nausea. This is an adaptive trait because it helps animals avoid potentially toxic foods. This form of learning happens with minimal exposure and can occur just after one pairing of a novel taste to see us with an illness, even if there's a long delay between the two. This readiness to learn associations quickly is an example of how evolution has shaped learning processes to promote survival. Okay, so just to wrap it up, this is our last slide here today. Drug tolerance and drug cravings as conditioned responses. So drug tolerance develops when the body becomes used to a drug, requiring larger doses to achieve the same effect. Over time, repeated drug use leads to physiological adaptations, and this can also be understood through classical conditioning, where drug tolerance and drug cravings are seen as conditioned responses. When a drug is repeatedly paired with a specific environment or set of cues, the body learns to anticipate the drug's effects. Just like if an alcoholic, somebody that drinks too much goes to a bar, they start to anticipate those effects. And so those physiological changes start to occur in the presence of these cues even before they have their first drink that day at the bar. This explains why drug cravings often occur when individuals are exposed to environments associated with the drug use. Siegel's research with rats, we have Shepard Siegel, and he conducted landmark studies on drug tolerance and the role of environmental cues and drug effects. Just to provide a quick overview of the study, Siegel gave rats morphine injections in a specific environment, and saline, which is for the control subjects, injections in a different environment. Over time, the rats developed a tolerance to the pain-relieving effects of morphine, but only in the environment where they consistently received the drug. Environmental cues like the room the rats were in became the conditioned stimuli that triggered these compensatory conditioned responses, the physiological changes that opposed the effects of morphine. In the familiar drug taking environment, these compensatory CRs reduced the drug's effect, leading to tolerance. However, when rats were given the same dose of morphine in a novel environment, they were more likely to experience an overdose because the compensatory responses were weaker or absent. This research highlights how drug tolerance and overdose risk can be influenced by contextual cues. When drug users consume the same amount of drug in a new or unfamiliar environment, the compensatory CRs may be weaker or absent. And this lack of anticipatory physiological responses means the body isn't prepared for the drug's effect, and that leads to a higher risk of overdose. So, for instance, if an individual who regularly takes a certain dose of a drug in a familiar environment may not experience compensatory responses when taking the new drug in a new location. Without these opposing physiological changes, the drug has a stronger effect, which can lead to an overdose. There's something called SOP, which is back to Wagner again. It's Wagner's sometimes opponent process theory, or SOP for short. And so the SOP theory is a model of classical conditioning that builds on earlier opponent process theories. SOP theory states that stimuli can sometimes elicit opponent responses under certain conditions. And so in the context of drug use, the drug's primary effects are like an A process, and the opponent responses is the B process. They're conditioned and help maintain homeostasis. These opponent responses become stronger over time due to repeated pairings of the drug, the unconditioned stimulus, with specific cues, which would be the conditioned stimulus. And according to Wagner, opponent processes aren't always present in the same form. They can sometimes manifest only in certain contexts or under certain conditions, such as specific environmental cues associated with drug use. Okay, so that was a mouthful, right? I know there's a lot of intense information in this chapter and it can be difficult to understand. So I hope this is helpful and beneficial to you. And if you have any questions, you guys know how to reach me. And I hope you have a great day, weekend, year, whenever you're listening to this. Take care.